Good. So, okay, please. Thanks so much. Okay. So what I would like to do is, and I have been asked to do it, is to emphasize why academic people and industry people should work more closely at the very beginning. Because, I'm a professor at the University of Illinois, but I co-founded 2 companies, so I have some experience in terms of trying to bring a product to the market. So I actually, the first reason why industries should approach the academe is because academic people don't think like industrial people. See, industrial people have a goal, let's say, to bring anti-cancer drugs. So if I go and join Pfizer or SmithKline or whatever, they will say, develop an anti-cancer agent, and I will look for new targets and go through the same process. It's called high throughput screening, get hits, get lead compounds, and so on. Academic people don't necessarily work that way. I had no interest in cancer, but I was forced into it, and I found out that there are some bacteria that live in our body for a long, long time. I was interested in microbial infection, bacterial infection. I had nothing to do with cancer. But then I found out that some of these bacteria that can actually go into human body and live there for years without doing any harm to the cell. So that's called a symbiotic relationship. When they live in our body, they consider our body as their habitat, and they would like to protect it from outside invaders. So these bacteria, one of them is called Pseudomonas aeruginosa, that infects cystic fibrosis patients. I found out would produce very intelligently designed proteins that will go specifically to cancer cells and attack them by interfering in multiple steps in the cancer pathway. But that's not all. Because the bacteria have 3 billion years of evolutionary wisdom. They were the first life form to arrive on Earth. So when they design a weapon to keep cancers or parasites or viruses in check, and I'm talking about a single protein called azulin, which has anti-cancer, anti-HIV/AIDS, viral activity, anti-parasitic activity and all that. But we will concentrate on cancer. We found out that these proteins, I have 2 of them, and the other protein is produced by another bacterium called Neisseria meningitis that causes meningitis. So Neisseria can cross the blood-brain barrier to go to the brain meninges and causes inflammation. But when resident in the brain, and if there is a brain tumor, Neisseria doesn't like it. Pseudomonas aeruginosa doesn't go to the brain, so it doesn't care about having a habitat in the brain. But Neisseria does. So Neisseria produces a very similar protein like the Pseudomonas protein called azulin, but because it knows that it has to enter into brain tumors which are highly vascularized, so it's very difficult to go to brain tumors, they put a 39 amino acid extra epitope in its weapon. So it's a modified weapon. If you go to war, you carry multiple weapons. And the point I'm trying to make is that Neisseria, depending on its habitat, will produce a little different weapon than a bacterium like Pseudomonas. Okay. But what we found out was that these proteins can not only kill tumor cells, they are non-toxic because they only enter into cancer cells, not into normal cells, and they can prevent cancer. That's oncogenic transformation of normal cells to cancer cells. So what is it that I'm trying to tell you? I'm trying to tell you that I have a new kind of protein that nobody has that can enter into cancer cells, interfere in multiple pathways by which cancer cells grow, is non-toxic, and can prevent cancer. So this is an example. So we just wrote a review in a journal called ICRN Neurology, and if you look at the title, you would say that we are talking about glioblastoma multiforme, a deadly form of brain tumor. And if you look at the authorship, first author is Dr. Fialho, who is from Lisbon, Portugal. So European connection. The next few authors are from India, Salunke, Manna, Mahali, and of course I am from the United States. So you see, we are talking about international collaboration here. And then the letters, I get a lot of letters, and I'll show you one more letter and then I'll stop. So this is the letter from an Italian doctor. His mother had glioblastoma, deadly tumor, 12 months, if you are lucky. So he wrote to me saying, what do I do now and all that, just to impress on him the fact that people can have cancer wherever they live. Obviously, we know that. But you know, so the question you will ask me is, but will it actually work in human? So I found the company I told you, and the company approached the FDA, and the FDA said, don't use the protein, that's a biologic, use a peptide, because you can chemically synthesize it, and that's called a drug. So, and the next one. So we used a peptide, a 28 amino acid peptide from this protein called azurine, and this peptide was tested in 15 stage 4 cancer patients, according to the FDA regulations, and if you look, you will see that 11 male, 4 female, 7 melanoma, 4 colon, these are various cancers. The entry is IV bolus, so it has to be injected 3 times a week. But adverse event, none. There is no toxicity, unheard of for anti-cancer drugs. And if you look at the response, no toxicity at low concentrations. There were 5 escalating doses that the FDA advised, and you will see that at low doses there was no beneficial effect. But as we increase the doses, at higher dosages, 2 persons, these were patients where their tumors were not responding to any conventional drug. But at higher doses, 2 of them had partial regression of their tumors, and 2 of them had complete regression of their tumors. So you can see that these are very different kind of drugs, and because they can prevent cancer and they are proteins, the interesting question that you can ask is, excuse me, can we express them in our genome instead of getting IV injections? Can we actually express these proteins in our genome so that these proteins will be produced in our body and will protect us from cancer? It's non-toxic, it prevents cancer, it kills cancers. That's the kind of experiments we are going to do in the future, and that will raise a lot of interesting questions about what we call patent eligibility issues, what you can patent and what you cannot. Okay. But then, as I said, I get letters. So I got this letter from a physician in Fairbanks, Alaska. And this is what she said. She said, Mr. Chakrabarty, you know, I have a prostate cancer, high-grade prostate cancer patient. He was doing well. There was a catheter inserted near the tumor at the ureterovesicular junction. And that catheter got infected with Pseudomonas aeruginosa. Pseudomonas aeruginosa has that property. It forms biofilms on what's called indwelling medical devices. Okay, just 2 more minutes. So I knew that Pseudomonas aeruginosa produces all these protein weapons to kill cancers in check. She got the clinical evidence that catheter got infected by Pseudomonas aeruginosa, and she says, well, the patient was doing well. But physicians don't like a catheter in a patient being infected with a bacterium. So they try to get rid of the bacterium, and she says after months, we finally got rid of Pseudomonas aeruginosa from the catheter that was inserted near the tumor. And see what happened? The cancer started to spread. It went to the bones, it went to the liver, and she said, I was desperate. How do I save my patient? And I said, was that Pseudomonas actually protecting the patient? So she now wants the Pseudomonas back in the catheter. Imagine that. But it also tells you, it's an interesting clinical study. FDA not FDA approved, so I said, wow, bacteria have been, lot of people for the last 30, 40 years or more have actually been trying to use Salmonella, Clostridia and other bacteria by injecting them into the bloodstream. They will go to the tumor, 99.9% of them will go to the tumor and try to grow. At least that was the idea. But you cannot inject live bacteria into the bloodstream because that causes immune response, toxicity problems and all that. And I said, wow, if I can have a biofilm of Pseudomonas aeruginosa in a catheter and insert it near any tumor, those bacteria will fight the tumor. And in order to make sure that the bacteria don't come out of the catheter into the bloodstream, I can encase the catheter in a membrane filter with a porosity that will allow only proteins of certain sizes, 40, 50 kD, to come out. Those are the protein weapons that will come out, but the bacteria won't be able to come out, so there will be no toxicity. So we did an experiment. We said, will it actually work? So we actually took mice, 2 mice, both with MCF7, human breast tumor, which you can see. Okay. So on the top, you can see that if the mouse didn't have a catheter near the tumor kept on growing, of course. But then if there was a catheter with the P. aeruginosa biofilm, after 4 weeks, the tumor burst open because of necrotic lesions, and then you can visually see it. After another week, the wound healed and the mouse started to move and so on. Point of all this is that academic research can actually open up completely new fields that the industry may not get into or know about or whatever. And of course that allows a university professor like me to then start up a company and try to bring those things to the market. So that's the point I tried to make to you. I recently attended a meeting where the National Cancer Institute is trying to ask the same questions, and it was a closed meeting with 25 people, and you can see they are from various industries and academic in Europe and the United States and all that. So this is something which we are trying to do, and the next speaker, Dr. PLO, will talk about what he's trying to do in collaboration with his Indian and US colleagues.